short revision for your unit number 2, that is cell biology. One of the most important, most scoring and most predictable unit. By predictable, I mean because the questions, the topics from this particular unit are very much predictable, that from here the question would always be there. For example, one of the topic is the cell cycle and its transition point or the regulation points that we have every single year, either in the fact-based segment or in the experimental segment or in statement wise, or maybe a combination of two. You get a question from the cell cycle. Then question from microbial physiology are also getting asked very recently. Then the protein sorting part about the cladrine, the caveole mediated endocytosis, exocytosis and all those things. So for the proteins sorting segment as well, we are getting a lot of question. Followed by that, we have questions asked from the nucleosome concept as well. If you look into the previous years, you'll get to understand the nucleosome concept, specifically the zigzag model and your solenoid model gets asked in the examination a lot of times. Sometimes questions from transposomes also get asked in, right? In the recent paper, we have got questions from the lac-operon as well, right? So lac-operon, it's an overlapping topic with unit number 2 and 3, which is molecular biology. So we'll be dealing with the lac-operon, trip-operon, ARA-operon in the next one short revision part, right? If you have not attended or watched the previous part of biochemistry, so you can have it. It's available on the app as well, and it is available on the entire playlist segment, right? You have the entire access for the same. Today we'll be dealing with the cell biology, and these are the most yielding topics of all time. And the same thing can be used for your DBT segment as well, because in DBT also cell biology holds a good importance, right? Let's begin. The first topic that we have to revise is the cell cycle and its regulation part only. If I talk about cell cycle, what is cell cycle all about? So it is a process in which a cell is dividing into two of its next generation, either 2 or 4, depending upon whether it is a mitosis or meiosis, right? Mitosis is in which there is one cell dividing into 2 and exact copy number is being formed. Then second is meiosis. Meiosis is when you have one cell dividing into 4. This is your reductional division that we talk about, right? So we have various phases in the same. I hope that you have revised about mitosis, meiosis in your own revision part itself. If you have any doubt, let me know. I'll insist upon that particular topic accordingly in further lectures. Right now, talking about the phases. So if I talk about the phases of cell cycle, there are two main phases. One is called as the interphase, which is also called as your preparatory phase, followed by the second one is M phase, whether it is mitosis or meiosis, depending upon which cell is it working in. In the interphase, you again have 3 of the phases, the G1, S and G2. Sometimes the cell might exit temporarily from the cell cycle and enter into the G0 phase or the quiescent stage that we talk about. In the G1 phase, there is cell growth, organelle duplication, and the preparation for the DNA synthesis happening in the S phase, we have the DNA replication occurring, histone synthesis increases, right? Then in the G2 phase, you have the protein synthesis for the mitosis, centrosome duplication is completed. Then we have the M phase, the mitotic or the meiotic phase, depending upon whether it is mitosis or meiosis. If it is mitosis, you have prophase, metaphase, anaphase and telophase. If it is meiosis, it is meiosis 1 and meiosis 2. In meiosis 1, you have prophase 1, metaphase 1, anaphase 1 and telophase 1. In meiosis 2, you have prophase 1, prophase 2, metaphase 2, anaphase 2 and telophase 2. The prophase 1 is the most elaborate segment of all. In the prophase 1, you get to see the main process of genetic recombination occurring in with the help of the mechanism called as crossing over via five, like there are five different steps in it: leptotene, zygotine, packaging, diplotin, and diakinesis. In the case of your like meiosis, it is the packaging stage where I have my main genetic recombination and the crossing over occurring in. Because of this genetic recombination, there is something called as variation that occurs in the human race. Like if I talk about the condition, or if we take the example of humans, why or how do we have variation in the parents and the next generation? So this is with the help of variation, this is with the help of the genetic recombination. When the egg and the sperm fuses in together, so when this egg and sperm, when these gametes are formed, they are formed via meiosis, right? These sex cells undergo meiotic division to make up the haploid cells, right? So during this haploid cell formation, when the meiosis is happening, so in the case of mitosis, you'll have the exact copy number being formed, but in case of meiosis, exact copy number is not getting formed. We are having the copy number, or we are not having the exact same mother cell being there. This is a variated part, right? Specific, like some kind of variation is there. This variation is with the help of genetic recombination. And when this mother cell, like the egg and the sperm, fuse in together, when the zygote is formed, you have the ploidy level being sustained to and back again. In addition to that, you have a good variety, like a complete variation of the same. So some features of the next generation might look like father, might look like mother, but they have some of the new set of features as well that gets created, because of which you have all the variations in the entire human race, right? So remember about this. Remember in which particular phase does it occur, where in which phase do we have the genetic recombination and the crossing over happening in? It is the packaging stage of your meiosis 1 in my entire cell cycle, if it is undergoing meiosis. Then, because it's a very important mechanism happening in, you have one cell dividing either into two or dividing into four. So the copying or whatever the mechanism has to uh, take place, this entire segment is very important, right? You have a very good uh, importance being hold, because any change or any problem, any kind of mistake done in the copying can cause DNA to get to produce any wrong protein altogether. So for that matter, we have something called as checkpoints, right? What do we have? We have something called as the checkpoints being there. So in every stage, whatever is completed in the previous one is cross verified. For example, you have, let's say, this is your interface. So this is G1, S, G2, and then you have the M phase being there. So when the cell is moving from the G1 phase to the S phase, you have G1 to S checkpoint being there. That whatever was required, or whatever was supposed to be done in the previous segment, is it done or not? So for that, G1 to S checkpoint play part in it. Then from S to G2, in the S phase, the main segment of DNA replication occurs. So this is a very important checkpoint. So from S to G2 checkpoint, the DNA, everything is checked that whatever was supposed to be completed in the S phase is done or not. Then from G2 to M, now from G2 to M, it is a main transiting point, because the cell is entering from the preparatory phase into the main division phase. So from here, over here as well, you have G2 to M transition being there. Last checkpoint is like in the metaphase. In your division phase, you have prophase, metaphase, anaphase, and telophase happening in. So from your metaphase to anaphase, in the metaphase, there's a very important checkpoint, or there's a very important function to be performed, which is all the sister chromatids, they all arrange at the equatorial plate. So from metaphase to anaphase, when this transition is happening, the checking is done. The checking is done that whether or not all the sister chromatids are arranged on this equatorial plate or not. Are they properly segmented over here or not? And are all the kinetochores arranged to the spindle fiber or not, right? So this is checked in the metaphase to anaphase transition part, right? So for these transitions to happen, for these checkpoints to work properly, we have two set of proteins working in. You have these proteins working in, you have cyclins and you have CDKs, cyclin dependent kinases working in. So what are the various checkpoints? G1 to S checkpoint, it checks for the DNA damage, nutrient availability, and the growth factor. Intra S checkpoint, during that time also there is a minor checking up done in that DNA replication is working properly or not. G2 to M transition, it ensures that the replication is completed, checks for any damage and all these parts. Then we have spindle assembly checkpoint, so it verifies proper chromosome attachment to the spindle fiber. Then you have P53 tumor suppressor, so it is the guardian of the genome. It arrests the cells cycle and it induces apoptosis if by chance the DNA damage, if there was any, is not repairable, right? So that is also important. Then cyclins and CDKs. So in every phase you have a different cyclin, you have a different CDK working in, and where do they work, it is written down here, right? Then you have CDK activity formula. So CDK cannot work alone. CDK's full form in itself is cyclin dependent kinase, because the functionality of your CDK is dependent upon cyclin. Cyclin and CDKs, they make up a complex together in order to proceed forward in the entire functionality. So CDK active formula is, active CDK is when you have cyclin plus CDK minus the inhibitors that are there, because CDK, if it is present alone, it might have some of the inhibitors attached to it. So if I want to have my cyclin dependent kinase working properly, I need to have the cyclin, associated cyclin joined in, like in whichever phase the my cell cycle is progressing, that particular cyclin would be there, plus the CDK will be joined in minus the inhibitors, right? Then during your G1 to S transition, there is something called as retinoblastoma and E2F. Okay, retinoblastoma is an inhibitor of E2F. Okay, it does not allow the E2F to enter into the nucleus and help in or let the cell go from G1 to S. So during my transition, during my G1 to S transition, this retinoblastoma need to be phosphorylated. Usually with the phosphorylation, some of the like majority of the proteins get activated. But over here, with phosphorylation, the retinoblastoma gets inactivated. As a result, it leaves the E2F, or the E2F is set free thereafter, and E2F enters into the nucleus where it can help out in the activation of various transcription factors, for example, GUN, MYC, TUS, all of these things, which will help in the G1 to S transition. And retinoblastoma is very stubborn, so it does not get phosphorylated once in one single go. It requires two different sets of cyclins and CDKs to function in, which will then cause the initially partial and then complete phosphorylation, and then the G1 to S transition gets completed, right? Then from metaphase to anaphase, in the From metaphase to anaphase, over here we have something called as anaphase promoting complex getting activated, which works as a ubiquitin ligase that degrades the cyclin during the mitotic exiting part, right? Then what are the various stages in different stages? So in the prophase, you have chromatin condensation happening, nuclear envelope breakdown occurring, centrosome separation happening in metaphase. The chromosomes align at the metaphasic or the equatorial plate, and spindle checkpoints get activated. In the anaphase 1, sister chromatids separate and movement to pole, and anaphase 2, anaphase B, spindle pole separation and cell elongation happens in. Then you have the telophase. So it is, there occurs nuclear envelope reformation, and we have the chromatin decondensation happening thereafter, right? Sometimes in some of the books you'll get to see the anaphase convert dividing into anaphase A and anaphase B, or majority of the time we consider it together only, right? Then meiosis 1, meiosis 2. Meiosis 1 is reductional division. In this, homologous sister chromatids get separated, the homologous chromosomes get separated, and crossing over occur in the prophase 1. In which particular phase do we have it? Specifically the Paquetin stage of prophase 1, right? Which all stages are there? Leptotene, zygotene, Paquetin, diplotene, and diakinesis. So in the Paquetin stage, we have all of these things occurring in. With the help of crossing over, you'll have genetic recombination done thereafter. Then meiosis 2 is equational division. In this, the sister chromatid separation will occur, and you will have 2n to n ploidy level getting completed. Then we have synapses. So during the genetic recombination time, during your crossing over time, how does it occur? So let's say this is one of the chromosome, and this is the second chromosome. So when you'll have the crossing over or the genetic recombination occurring over here, you'll have a chiasma formation, somewhat like this. It's one of the color would be here, other would be here, like a cross. So it's like this happening in. So this is the chiasma formation, which is the physical manifestation of the crossing over events, right? So we'll be discussing about, like, this is very, very important, right? Then we have the cell death mechanisms. So in this you have three of the segments majorly: apoptosis, necrosis, autophagy. Apoptosis is what? That every cell, whatever cell is there, okay, this particular cell, it does not live in our body for the entire life. Every cell has a specific life to it. For example, the red blood cells have a age of almost just 120 days. In every 120 days, the red blood cells keeps on getting regenerated, right? So after these 120 days, the cell needs to have a clarity, like the dead cells, whatever are there, they need to be removed from the body, right? This occurs via the programmed cell death, via your caspase cascade activation, right? Then you have necrosis, which is uncontrolled cell death due to injury or pathological condition. What is necrosis? Imagine that I get a injury here, or any person gets an injury on their hand or anywhere in their body. So the cells over here were not supposed to die off as according to a normal scenario, but because of injury, because of any problem, some cells died from here. So over here the mechanism of necrosis will begin in, right? So necrosis is what it is: uncontrolled cell death due to injury or pathological conditions, right? Then you have autophagy as well, which is your self digestion process for the cellular components being recycled. The next concept of the cytoskeleton. So what is a cytoskeleton all about? Cytoskeleton is nothing but the entity which provides the cell a complete framework. What is a framework? So for a cell to have its different components arranged properly, for its components to have a proper positioning to them, you have something called as, like in your body also, every single organ, every single, like your entire body gets a framework with the help of a skeleton, right? In the similar fashion, in my in every single cell also, we have a specific location to every entity, every pointer, every component being there. So that is what cytoskeleton is, right? Cyto is related to your cell and skeleton is the framework. So the segment which provides the framework to the cell, that is what cytoskeleton is. Now in the cytoskeleton, you have microfilaments, you have intermediate filaments, and you have the microtubules being there. Microfilament is made up of actin. If I talk about the structure of actin, so you have the G actin, which is the G actin, which is the globular actin. These globular actins joining together to make up the filamentous F actin, right? Then actin polymerization, so ATP actin, it adds to the plus end. So when from the globular to the filamentous part, when I have my actin filament getting formed, one side is called as the plus end, the negative side is called as, like one is called the plus end, one is called the minus end. Write down what is pointed end and barbed end. One of it is like plus and one of is negative. So which one is the barbed end, which one is the pointed end? Try to recall and let me know. Then we have the treadmilling. Treadmilling is continuous addition and removal of the globular actins on this filament that we have. So when the polymerization and depolymerization is getting done on a very, very regular interval. Then we have actin binding proteins. So proflavin and coflavin, like three main different actin binding proteins are there. One is called as the proflavin, one is called as the coflavin, and one is called as the tropomyosin. So functionality of proflavin is to polymerize, to add on the globular actin here. Functionality of coflavin is depolymerization, to remove the G actin from here, and tropomyosin is to make sure that the entire stability is getting done, right? Then we have the intermediate filament, abbreviated as IF. So the composition is that there are tissue specific proteins called as keratin, vimentin, desmin, neurofilaments, etc. IF structure, in this we have the 10 nanometer diameter, right? They are most stable cytoskeleton elements. They are not very much dynamic in nature. And last is about the assembly. So we have a monomer, then a dimer being formed, then a tetramer, then a protofilament, and finally a filament is being formed in, right? Then third is the microtubules made up of tubulin protein. The tubulin is alpha tubulin and the beta tubulin. So these are there occurs formation of heterodimers over here, and it makes up around 25 nanometers of diameter. So over here, MTOC, microtubule organization center. So over here, it nucleates with your gamma tubulin. Dynamic instability, again, there's a constant addition and subtraction. So a constant polymerization and depolymerization is happening in. And then over here, kinesin and dynein are the one that will be, these are the two protein that helps out or that works in the with the motor proteins, right? So if I have microtubule, remember kinesin and dynein, they always move in opposite direction of your microtubule. In normal scenario, kinesin always moves in positive direction. This is the standard behavior of kinesin protein. Dynein always moves towards the negative direction. Now my microtubule will always be moving in a opposite direction of the protein that is there. So if kinesin protein is there, you will be having the positive end to, like kinesin moving toward positive. So microtubule will move towards negative. If dynein protein is there, so my dynein will always move toward negative, which is the normal scenario, and my microtubule will move toward the positive end. So remember about the basic nature. Kinesin always positive, dynein always negative, and microtubule will be moving always in the opposite direction of the protein that is participating in. So you can get an experimental question from this part. Then we have cytoskeleton element. Three different are here, which we have just discussed. So microfilaments diameter is around 7 nanometers. Composition is, it is made up of actin, and main function is the stem movement and your in contractibility. Intermediate filament diameter is 10 nanometer. Many proteins are there, and structural support is the main function. Then microtubules, 25 nanometer. Tubulin is the protein that is helping out, and the main functionality is intracellular transport and mitosis. Then I have the next concept of nuclear pore complex and your nuclear transportation. Again, very, very important and very scoring topic, because you get questions from the nuclear transport very easily, and from the nuclear pore complex as well. This is very important. Like the structure of nuclear pore complex is very, very important. So if I talk about the nuclear pore structure, so it's a 8-fold symmetrical structure having approximately 120 MDA as the weight, and it is composed of nucleoporins. Nuclear envelope, if I talk about the structure of my outer membrane of my nuclear envelope of my nucleus, it is a double membranous continuous with your endoplasmic reticulum, and it contains the nuclear pore for the functionality, right? For the transportation and all those parts. Then we have the RAN GTP gradient. So you have two different segments. One is RAN GTP, second is RAN GDP, right? So RAN GTP is the one which will be helping in, like the GTP and GDP gradient helps out in the entire nuclear import and the export part. So let's say this is the nucleus and this is the cytoplasmic region. So inside the nucleus, you will be having more concentration of RAN GTP, and outside over here, you have more concentration of RAN GDP, and you will have a proper movement of the molecules occurring in, right? Then if any protein needs to be moved from one of the location to the next location, right, and the next location is nucleus, this protein need to have a specific signal to it, or it need to have a tag to it. This particular tag is called as your nuclear localization signal, which is your NLS, which is having, like, it is a basic amino acid cluster that is required to target any protein towards the nuclear region, right? Write down what is or which two amino acids are majorly seen in the nuclear localization signal. I think this particular question was asked in the examination also. So write down which two main basic amino acids need to be there for any specific signal to work as a nuclear localization signal, right? Then we have transport mechanism. So you have the important as the class of protein that is helping in out, right? So important alpha beta pathway. So this is the normal classic import receptor, import and export mechanism which occurs. In addition to that, we also have exportin 1, which is CRM1, which is the main nuclear export receptor, which is leucine rich NES, nuclear export signal recognition. Then you have RAN GAP, which is cytoplasmic RAN GTPase activating protein, and RAN GEF, guanine exchange factor,